Hello, my name is Jenna Philippon, and today I'm going to be presenting on genetic and developmental disorders. I have attached a PowerPoint presentation so that you can follow along and take notes while I present. The objectives of this presentation is to help you understand the following: human cells, chromosomes, and genes. I would like and hope that you're able to explain the errors that occur during meiosis and mitosis. Explain the differences between autosomal recessive, autosomal dominant, and X-linked disorders. To understand inheritance patterns in single gene disorders, which are seen through geo-imprinting, anticipation, and mitochondrial inheritance. And to understand the environmental issues can also cause problems with development. What is a genetic disorder? A genetic disorder is a disease caused by an error in individual's DNA during fetal development. These errors can include error in cell division, an error in chromosome division, adding or deleting chromosomes, mutations within the gene, or a single gene. Genetic disorders all start in the cell. There are two key parts: the nucleus and the cytoplasm. The nucleus is where chromosomes and DNA are organized into genes, which are responsible for our physical attributes and body functions. These chromosomes and DNA organize into genes needed for life. The cytoplasm rids the cell of waste materials and releases energy. It creates all the required material needed to complete the task of development. Genes in the nucleus are dependent on the cytoplasm. The nucleus and the cytoplasm need to be working in unison so that there is no defects in gene development. If an error occurs in any phase during development, it opens a wide range of genetic disorders. Cell division. There are two types of cell division: meiosis and mitosis. First off, cells have 46 chromosomes, and each has 100 genes. The 46 chromosomes are really 23 complementary pairs. The 23 chromosomes come from the female, and 23 chromosomes come from the male to make a total of 46. 22 out of our 23 chromosomes are autosomes. The last chromosome, the 23rd, is a germ cell, which is either an X or Y chromosome, which helps ID gender. The X is given by the female and the Y is from the male. So XX is a female and XY is the male. During prenatal development, cells divide two ways: in mitosis and meiosis. During mitosis, this process is seen in all cells, from skin cells to liver cells, and the rate that this process occurs depends on the individual's age and the location of the cell. For example, if we have a cut on our arm, it's going to take less time to heal than having an issue with our liver or a problem within an organ in our body. And the younger that we are, things will heal a lot faster than if we were older, elderly individuals. Meiosis, on the other hand, mitosis is when two daughter cells, 46 chromosomes each, are formed from one parent cell. Meiosis is when 4 daughter cells, 23 chromosomes each, are formed from one parent cell. This is only seen in the germ cells, and these cells create the sperm and the egg, giving those X and Y gender identities. The differences in mitosis and meiosis are usually seen in the first phase of cell division. This is when genes cross over. This is a very important part of cell division because it makes sure and ensures that we don't have clones as siblings. The genes cross over to give us different genetic identities. Having errors at this phase is rare, but it does happen. Cell errors occur in chromosomes. Division errors are seen as non-disjunctional. This is when one sperm or egg gets 22 or 24 chromosomes, not the required 23. The error causes a defect like Down syndrome or Turner syndrome. This error is usually seen in chromosome 21, which triggers the Down syndrome. This error occurs both in mitosis and meiosis, but more commonly seen in meiosis. This error in division causes there to be one cell that receives 22 chromosomes, which will die, and one cell which will receive 24 chromosomes, which can sometimes live, resulting in Down syndrome. 95% of Down syndrome occurs this way. However, it can also be caused by translocation and mosaicism of chromosomes. Children with Down syndrome or Turner syndrome, 2 of these errors that can happen, will suffer from intellectual delays, unusual facial features, congenital malformations. This usually occurs in 6 to 9 out of 1,000 live births. And this is how 95% of individuals with Down syndrome will be, this is how it will be caused. There are type of errors in chromosomes. Chromosomes can be lost. This is commonly seen in Turner syndrome. This syndrome only affects girls. The only disorder that the fetus can survive without a chromosome. The fetus will only have an X. They are missing an extra X or Y to make them have an XX or XY, total of 46 chromosomes. They're missing that one. Some of the side effects of this is visual impairments, perceptual impairments. They'll have a broad shield chest and wide nipples. But 99% of these children, it will be caused as a miscarriage. So it's a very rare syndrome. It is actually very common to Down syndrome, but there is not as vast as an intellectual delay. Mosaicism is when there is a loss of an extra chromosome. It is very rare and seen only in 5 to 10% of chromosomal abnormalities. Translocation is when seen in cell division between mitosis and meiosis phase when chromosomes break down and exchange with others. There is a transfer of chromosomes, but to completely different ones. So chromosome 14 will attach to chromosome 21 instead of the other 14, which it is supposed to attach to. This error causes 46 chromosomes, which are needed for development during fertilization. However, it results in Down syndrome because there's 2 chromosomes 21. There's 21 in chromosome 14, 21. So this trisomy, which is 21, which is when there is 3 chromosomes that are labeled as 21. This 3 chromosome setup is the common reason for Down syndrome. There are more types of errors in chromosomes where things can be deleted. This is rarely seen in cell division. Deletion occurs at the molecular level. One example is triducat, which is the deletion of the 5th leg of a chromosome, which is a common deletion. And this causes seen as a high-pitched cry. Williams syndrome is a microdeletion, and it is a continuous gene syndrome caused by the deletion of a number of adjacent genes. With recent improvements in technology, this is easier to detect due to the use of FISH, which is fluorescent in situ hybridization. Deletion is not usually seen as losing all of a chromosome, but more of a part of a chromosome. It happens in 1 in 50,000 births, and it can cause unusual facial features with round face, widespread eyes, lower set ears, and intellectual disabilities. There is another disorder that can be caused along with Williams syndrome, and that is YCFS. And this causes children to be born with cleft palates, heart defects, abnormal facial features, and learning disabilities. About 10 to 15% of all births had abnormalities, and 95% of all chromosome abnormalities do not survive 2 terms. So if there is an abnormality, it is very rare that they will live throughout the whole process. The main issue in chromosome disorders is that there are too many or too few genes, which causes an additional or deleted chromosome. So genes can be turned on and they can be turned off. First off, we need to know what the function of a gene is. I just mentioned gene, but what is the function of the gene? The gene's function is to produce certain protein like chromosomes, blood type proteins, and enzymes. It is important to regulate the development and function of the body. Genes are composed of 2 strands of various lengths of DNA which bond together to create the double helix. This double helix formation is considered a chromosome. The DNA is made up of phosphorus and sugar molecules on the outside and 4 chemical nucleotides on the inside. Very much of what you might have learned before. You have your cytosine and guanine and adenine and thymine. The A matches up with the T and the G matches up with the A. This creates different gene combinations, and there's actually over 3.3 billion gene combinations that can be made. It is important to remember that not all genes are turned on or off at the same time, and that they're only active, like some genes are only active during fetal life and some are never active, and we never want them to be turned on or become active. Errors occur when the body does not properly turn the genes on or off. Gene errors and disorders can be caused by transcription, translation, mutations. There's a couple of different types of mutations where it can be a point mutation, an insertion, or a triple repeat disorder. Gene disorders can be caused by selective advantages, single nucleotide polymorphosis, which is SNPs, single gene disorders, which is autosomal recessive disorder, autosomal dominant disorder, and X-linked disorders. Let's discuss transcription and translation first. Transcription is when you unzip in the DNA formation to create a template for the formation of mRNA. mRNA is messenger RNA, which helps transport information across the cell. When the DNA unwinds, it helps create a template for mRNA. mRNA has all the same nucleotide bases, besides thymine, which instead it has uracil. One would think it would be a commonplace for an error, but unfortunately there is a proofreading enzyme set to catch and repair any problems that do occur during this phase. Translation. After transcribing the DNA, the mRNA goes from the nucleus of the cell to the cytoplasm so proteins can be produced. This process is called translation. At this time, mRNA attaches to ribosomes, which read the three-letter code called codons, which are made up of the amino acids. These amino acids are building blocks for proteins. This is kind of like the process of when a DVD is read in a DVD player. It's able to read the codes on the disc and play them through the TV. Once the codons are read, tRNA comes in and carries the codons to the ribosomes, creating a protein, which is then released into the cytoplasm. The protein will be used within the cytoplasm and then released into the bloodstream, or it goes into the Golgi apparatus where it can be released into the body by the cell membrane. Gene mutations. One of the gene mutations is called point mutation. Any error in translation process affects the protein creating an abnormal protein. This error in protein development can cause gene mutations. Once a gene mutates, it cannot change back to normal. The mutated gene then becomes part of an individual's genetic code, which can later be passed on from generation to generation. Point mutations are a common type of mutation, and it is when there is a single base pair substitution. One section substitutes and goes to another's part. It is not always a problem, but when it does occur, these type of mutations can cause missense mutations or nonsense mutations. Missense mutations is when there is a change in the triple code of DNA, which subs a different amino acid in the protein chain. An example of this would be PKU, which is an unborn error in metabolism. This is caused by the body to be unable to metabolize amino acid phenylalanine. This inability can cause the body to have a buildup of this amino acid, which can later cause brain damage. Nonsense mutation is when there is a single base pair substitute, which creates a stop codon and prevents protein development. It actually terminates the protein. An example of this would be neurofibromatosis. This is when neurofibromin, which is a tumor suppressor, is not formed, which causes benign tumors to develop in the body and the brain. This can cause children to have higher chances of ADHD. Insertion and deletion are other types of gene mutations. Mutation is caused when nucleotide bases are added, deleted, or even shifted. And this can be seen as a frame shift. The most common insertion is seen as spinal muscular dystrophy resulting in polio. A common deletion is in Duchenne muscular dystrophy. Additions or deletions of genes can lead to a frame shift. This is when 3 base pairs reading a frame is shifted, which causes a non-functional protein. And an example of this would be Tay-Sachs disease. A triple repeat expansion is seen for an unknown reason. There is an overproduction of codons in a certain region of a normal gene. This triple repeat can cause a gene to turn off, leading to Huntington disease or Fragile X syndrome. Selective advantage. The number of instances in genetic diseases depends on the amount of mutations that occur on the amount that are removed. Natural selection helps eliminate these problems. For example, individuals with sickle cell do not live as long as their counterparts, which means there is a less likelihood that the genetic code will be passed on to the next generation. So the selective advantage is showing that by evolution and people not living as long, the genes aren't having a chance to be passed on, so the errors are being eliminated. There are many single gene disorders that we will now discuss. The autosomal recessive disorder is the first one we will discuss. But before we start discussing disorders, let's hear some facts. People of all races and geographies share 99% of genetic identity. I found that kind of crazy. This means that there is only 0.1% that we do not share. This 0.1% is comprised of over 3 million single nucleotide polymorphosis. That means we're almost all the same besides a 0.1% of our genetic makeup. The single gene Mendelian disorders. George Mendel was a scientist who studied genes and later discovered dominant and recessive genes. He did this by studying pea plants. He had one green and one yellow plant and created a hybrid plant that was green. He discovered that both genes are inherited, but the dominant gene is not always inherited, but is also seen as opposed to the recessive gene, which is still inherited but not readily seen. These traits are inherited recessively and can become visible. And this happens in 1% of the population. 1% of the population has that recessive gene that is inherited that becomes visible. Mendelian disorders can occur as seen in 1% of the populations that it occurs with. And these can be seen as autosomal recessive or autosomal dominant or X-linked disorders. Autosomal recessive disorders. I added the picture so that you could easier understand how this is all occurring. In the 1700s of Mendele's disorders, 1,700 of Mendele's disorders are autosomal recessive, which means they are inherited from the mother and father. For example, Tay-Sachs, which is a progressive neurological disorder, can be caused by the absence of an enzyme, which prevents the proper breakdown of nerve cells, which means that they are unable to be metabolized. This later causes toxins to build up in the brain, later causing brain damage and early death. This is caused when two genes, normal being the dominant and abnormal being the recessive, the non-dominant gene, The genes get combined through fertilization as an egg. If you look at the picture, this will better be easier to understand. Two things that can happen to the egg during fertilization. The egg can be fertilized with homogeneous combinations, meaning that they will carry the same ally, or they can be fertilized with heterozygous combinations, meaning that they will carry different genes. If you look at the picture, you can see the different scenarios. The last scenario shows you how two recessive genes can combine and cause a disorder. Autosomal dominant disorders. 4,500 autosomal dominant disorders have been identified. It occurs in about 1 in 500 live births. It differs from autosomal recessive disorders in many ways, which include incidence rate, characteristics, and mechanisms which create the disorder. It is caused by a single gene abnormal ally. If you look at the picture, you can see that there is only one infected gene, and that gene is the dominant gene. When normal and abnormal, dominant and recessive genes combine, the autosomal dominant gene shows. This disorder is equally as frequent in both genders. An example would be arachnoidolysis and neurofibromatosis. Another type of single gene disorder is X-linked disorders. This is a recessive disorder caused by a mutation on the sex chromosome. The actual X chromosome becomes mutated. This disorder primarily affects males because they only provide with one chromosome, that is the X, and not given the Y. This causes disease. There are over 900 X-linked disorders, which include Duchenne's muscular dystrophy and Fragile X syndrome. The muscular dystrophy is caused by a mutation in the muscle cell membranes. It causes the muscles to become weak, and individuals will end up being wheelchair bound. The muscles of the heart and the diaphragm also are affected, and that can lead to an early death. The brain development is also impaired because the muscles within the brain are also infected. So this affects all muscles of the body. Fragile X syndrome is also a very common X-linked disorder. There are about 25% of males and 10% of females who have intellectual or learning disabilities are actually affected with the X-linked condition. Revising Mendele's genetics. Genomic imprinting. Unlike Mendele's belief of phenotype, characteristic types from mom and dad are passed on to the next generation. This is more of a phenomenon because the traits are inherited from the mother and the father, but there is a gene deletion that occurs from either the mother or the father. For example, if the long arm of a 15 chromosome from the father was deleted, the child would be born with Prader-Willi syndrome, which is characterized by short stature, obesity, intellectual disabilities. However, if it occurs on the mother's side, the child will be born with Angelman syndrome, which is characterized by intellectual disabilities and epilepsy. The reasons for genomic imprinting remains unclear. Another thing is when you see revising Mendele's genetics is anticipation. As Mendel said, genes are inherited and shall look the same from generation to generation in a given family. In this instance, The abnormal genes manifest, increasing the severity of the disease or disorder with each generation. An example is Huntington's disease, which is caused by a triple repeat in the gene formation. It is autosomal dominant progressive neurological disease, which associates with the movement and cognitive disorders, impairments which increase with each generation. Mitochondrial inheritance is another issue with revising the genetics. Cells are filled with mitochondria within its cytoplasm, which is responsible for maintaining the cell's function by oxidative phosphorylation. These organelles have DNA which is different than nuclear DNA because it is in the circular formation instead of the double helix. The genes within the DNA can mutate, causing the mitochondria to not perform efficiently. This deficiency causes deficient energy production and disease. MELIS, which stands for mitochondrial I don't know how to say that word, lactic acid in stroke-like episodes. It is characterized by stroke and dementia. If you look on your PowerPoint, it will tell you the name of that word. I just struggle pronouncing it. It is a disorder that is carried by only female genes, only since the egg contains mitochondria and cytoplasm. An unaffected mother is able to pass the deficient gene on to her children, both male and female. Males can be affected, but they cannot be carriers. Other mitochondrial disorders can cause blindness, muscle weakness, or deafness. There are over 60 mitochondrial disorders that have been identified. The environment. Environmental influences. The environment can affect the phenotype of an individual's gene. The phenotype is the way genes are expressed, as opposed to the genotype, which is the genes a person has. The environmental factors can affect the way genes are expressed. For example, the book was described a second generation Asian who is growing up in the US. Their offspring are going to be taller due to the increase in protein intake during adolescence. On the other hand, some disorders like diabetes, cleft palate, are affected by both the genotype and phenotype factors. Some individuals are predisposed to have specific defects. However, if unaware and untreated early, they can have different outcomes. To summarize, humans have 46 complementary genetic chromosomes which determine our physical appearance and biological makeup, but affect what we pass on to our offspring. When genes mutate, split incorrectly, delete, or add chromosomes, it can have a lasting effect on the outcome of a person's offspring. Many problems can occur during pregnancy. However, with all the issues that occur, 95% of infants are born without defects. To review, I hope by the end of this presentation you're able to understand what a human cell is, chromosomes, and genes, what errors can occur in meiosis and mitosis, what are the differences between autosomal recessive, autosomal dominant, and X-linked genetic disorders, what is the inheritance pattern of a single gene disorder, and what the environmental factors can also cause issues within development. Some interesting information that I found were that females have over 2 million eggs. Trisomy is a common cause of miscarriages. This is when we talked about before with Down syndrome. Chromosome 21, which is commonly seen, has there's 3 of them. Human genomes contain 20,000 to 25,000 genes. where a fruit fly contains 13,000 and a roundworm contains 19,000. I find that kind of astonishing, considering that I feel that we are more complex than those two species. Chimps share 99% of our human genome. People of all races and geographies share 99% of the same genetic identity, and there is only 1% that we do not share. I found that to be unbelievable because we are almost all the same. Many problems can occur during pregnancy, however, with all the issues that can occur, 95% of infants are born without defects. We come to the end of my presentation, and I have left the references for you. The information for this presentation has been provided from our textbook in Chapter 1, if you need to reference any of the information. The pictures have all been referenced with their links provided in their websites. If you have any questions, please leave them for me, and I hope you enjoyed viewing my presentation.